Hello, everyone, and welcome to Vesicular Transport, General Biology. This is an extremely important topic to have learned and understand before you get more into the cell. And vesicular transport, when we think about a vesicle, it's just a tiny little ball of, it's called a liposome structurally, but it's a ball. It's a ball surrounded by a lipid bilayer. Now, there are two different types of transport, vesicular transport. You use vesicles to transport different molecules, and there's one called endocytosis, and there's another called exocytosis, what we'll talk about. Now, the transport of extracellular fluid contents into the cell is usually called collectively endocytosis. It's used for cellular gain, so the cell gains things. So you can kind of see how it's getting things from the outside. It's usually a transport of fixable energy into the cell for food. Subclasses of this type of vesicular transport include penocytosis, phagocytosis, and receptor-mediated endocytosis, and we'll go into that. So here are the subclasses of endocytosis. So turning your attention to here, this left side with the lasers. Say this is a food particle, and we have phagocytosis, where your cell's cytoskeleton, which we'll talk about later too, is going to build kind of these two little finger-like projections called pseudopodium, and they're going to wrap around this food particle, and they're going to fuse these two membranes together to make a vesicle. And that's going to be called a phagosome, or a food vacuole. And that's going to be their food. And then, like we talked about earlier, the lysosome is going to merge with that food vacuole and digest whatever is in there. The lysosome is just this vesicle contains all the digestive enzymes. Now, penocytosis, we have extracellular fluid here. Peno meaning drink. We'll say it needs some ions from the extracellular fluid. Well, the cell is just going to kind of fold inwards and then make into a vesicle. And that would have all the nutrients it needs. Now, last we have a receptor-mediated endocytosis. Well, we have a signaling molecule that attaches to a receptor, and it tells the receptor to begin endocytosis prematurely. Or not prematurely, but rather, it signals for endocytosis in that particular area. So a receptor would kind of merge with it. It's called a ligand, receptor-ligand complex, I believe. And it would kind of signal the cytoskeleton to pull down and wrap around that ligand. Alright, now exocytosis. It's used to secrete an intracellular product. It can have some function in endocrine signaling in humans. So, for example, a signaling molecule would be secreted from a specific area in the human body. It can be used to excrete cellular waste, such as in a paramecium cell. I actually have a video of that on my channel. And it can also be used defensively to secrete toxins, such as leukocytes or white blood cells in your body secreting different species or toxins to kill invaders. Alright, now that is it. There's no subclasses for exocytosis, actually. There's one more thing I wanted to turn your attention to. Well, can you secrete vesicles within vesicles? Absolutely you can, and they're called microvesicles. You can secrete vesicles outside, or inside too, but it's more of an advanced thing. But this is pretty much what you should be able to memorize, these types of vesicular transport. Anyways, I appreciate you guys spending your time here, and I hope this helped. Goodbye.